And the next one is going to be by Hans Clevers. And I think if you read back through his journey through science, what you see is somebody who really didn't anticipate where he was going. He was an immunologist and couldn't know that he was going to end up a Wnt signaling person. And I think we're in for a really wonderful story. And I'm just going to turn it over to you, Hans, and let you take over. Thank you.
Okay, thanks very much, Stan, for a brief but robust introduction. Let me first start by thanking although they're not here, the founders of the Breakthrough organization for this wonderful prize and quite an incredible event yesterday evening for the ones who participated there, and also UCSF for hosting us today. I only made one contribution to UCSF, which is one of my first PhD students, Jeroen Roze, who I think is in the audience, who's been a faculty here for 7 years now. I'll be talking about the gut. You see the small intestine here. These protrusions into the lumen are called villi. We have been studying these little structures that are called crypts, the crypts of Lieberkühn, and these are the homes of these incredible stem cells that fuel the fastest self-renewing process in the mammalian body. Everybody's presented some history. Mine goes back a few centuries. The crypts of Lieberkühn. Lieberkühn is a German scientist. I found out that he actually studied in Leiden in Holland, discovered his thesis. Now my PhD students complain a lot about writing theses. This guy actually wrote his thesis in Latin. So I took Latin. This is now 45 years ago, I think, at high school, 40 years ago. Didn't help me at all to read this, so I then turned to Google Translate, and I could quite easily resolve that actually what he did is he infused hot wax into tissues like the gut, then digested away the live tissue, and ended up from the inside structures that really we now know are the villi. So you see the blood vessels here, and if you flip this around, these little indentations that you might see in between the blood vessels are the impressions that the crypts make towards the outside, so the muscle layer of the gut. So this is why we term these structures crypts. Now this is how we now view this system. A villus is surrounded by about 10 crypts. Every crypt houses stem cells near the very bottoms, and they were unknown for a long period of time. We know that every crypt produces 200 to 300 cells per day. A mouse has 1 million crypts. We have more than 1 billion, which means that we produce about 100 to 200 grams of cells every day in our gut. It takes 2 days from a cell while it divides to go from here to here, the crypt to villus junction. Another 2 days as a differentiated cell, it'll travel up to the tips of the villi and it'll go apoptosis. So a 4-day self-renewing tissue. Now neutrophils live for 3 days, but almost all other cells in the body live much, much longer. So we thought it was a good system to start studying adult stem cell biology, We cloned TCFs a long time ago when I started my lab as T-cell factors. There's 4 of them. When we knocked out TCF1, the mice have very little thymus. We didn't really know why. Much later, 96, 97, we realized that the TCFs are at the end point of the Wnt signal transduction cascade. I won't say too much about it, but we have about 20 or so Wnt's encoded in our genome, small secreted proteins that are lipid modified. They use frizzled and LRP5 and 6 as receptors. There's a a destruction complex in the cytoplasm of a cell. A prominent protein there is APC, defined amongst others by Bert Vogelstein as a tumor suppressor in the gut. In the off state, TCF represses transcription. In the on state, it activates transcription. Wnt signaling was really only studied in development. We knocked out the TCFs and looked for the functions in adults, and we found that if we knock out TCF4, mice are born normal, but they have no crypts. So it looked like the major driving force behind this self-renewal process and behind these stem cells was Wnt. And that actually has been confirmed many, many ways in the gut, but also in many other adult organs, that Wnt is probably the major driver of self-renewal in these tissues. At about the same time, we got interested in colon cancer. We read that in the absence of APC, beta-catenin is very high. I then got a collect phone call from Baltimore. This was Bert's secretary, Bert Vogelstein, who proposed a collaboration. And we actually collaborated together, and we shown that in colon cancer, you indeed find high levels of beta-catenin and TCF in the nucleus of these cells, and the situation is very similar to what you would see in a cell that undergoes active Wnt signaling. You're activating some program, and it was not too difficult to propose that the biology of colon cancer was very similar, if not the same, as the biology of the normal stem cells in the gut. What we did then, we had to wait a few years till Pat Brown invented microarraying, and we determined the gene program that's activated by TCF in the gut. Most of the genes are expressed in these rapidly proliferating transient amplifying cells. Some of the Wnt target cancer genes are expressed in these big Paneth cells. These are the cells that sterilize the lumen of the gut, was known for a long time. And much to our surprise, one of the genes, LGR5, lit up tiny little cells that we and almost everybody else in the field had missed. And you can see the size difference between these large Paneth cells and the LGR5 cells. Somewhat surprisingly, if they were the stem cells, we would have expected them to be quiescent, and we never find that these cells stop cycling. They have a cell cycle time of about 24 hours. They were actually seen by Charles-Philippe Leblanc, a sort of forgotten stem cell pioneer. He worked with the Curies in Paris, learned to use radioactivity in biological systems, defined Golgi. He defined that every cell in the human body or in the mouse body makes protein. It was believed that only liver makes protein. And he also used labels for DNA and defined progenitor cells, stem cell compartments, and differentiated cell compartments. He was still active at the age of 94. He passed away quite recently. Gave a PowerPoint presentation, his first, and he remarked, "A month ago I thought PowerPoint was a tool for sharpening pencils." So now I'll try to summarize what LGR5 does. We only found this out, we and several other labs, in the last year. So what you see here is frizzled and LRP5 or 6, which together can bind one of these 19 Wnt's. This leads to the phosphorylation of the tail of LRP, stabilizes beta-catenin. The nucleus starts transcribing TCF target genes, and amongst those are these transmembrane E3 ligases called RNF43, ZNRF3. They will ubiquitinate frizzled and, in a negative feedback loop, remove the receptor complex from the surface. So what this does is actually you have a brief Wnt signal during development, and then the signal basically is extinguished. LGR5 has a function here, because if you do have LGR5 and you have its ligand, R-spondin, R-spondin can actually bring together RNF43 and frizzled. So as a decoy, it now moves away the E3 ligase, goes inside the cell, and frizzled and LRP that are bound by Wnt will now, as it's stated here, result in a prolonged and robustly enhanced Wnt signal. And we since, and other labs have seen this, R-spondins are actually very potent stem cell growth factors, and they are these potent growth factors because they can, a small Wnt signal is, by the activity of R-spondins, is prolonged and robustly enhanced because the receptors with their ligands stay on the surface of these cells for as long as they apparently want. Now, to study if, this is now back a few years, to study if LGR5 is indeed a stem cell marker, if these Leblanc cells, as predicted by Leblanc, are the stem cells. We made a number of knock-in mice. This was done by Nick Barker in the lab, who is now in Singapore. The best one is this one here. We knocked in green fluorescent protein separated by an internal ribosome entry site, so we can produce two proteins of one mRNA. We have a Cree that's inducible by tamoxifen, a fusion with the estrogen receptor. This mouse works fantastically well. So we see in the gut, but in many other tissues, we see small cells that are green. And in all the cases that we or other labs have studied these cells, they are the stem cells of that particular tissue. I hope you can see this. 3 villi, 6 or 7 crypts, and in every crypt, the Leblanc cells now light up in green. So the green cell should have the inducible Cree-ER, and when we cross this mouse to a Cree reporter, for instance Phil Soriano's reporter, and then activate it in an adult mouse with a low dose of tamoxifen, we mark stochastically a single of these putative stem cells in blue, lean back, and then over the next few days we should start seeing rapidly proliferating daughters. By about 2 to 3 days, the first blue daughters should start to differentiate out. We know there are about 5 or 6 cell types in the gut, and if this cell is multipotent, which it should be, we should see all of these cells lighting up as blue cells. So then from day 3 towards day 4 or 5, the cells are now exposed to the lumen of the gut. very hostile biological environment. They help us digest and take up nutrients into the blood and lymph vessels, and by day 4 or 5, they reach the tips of the villi and they undergo apoptosis. And this summarizes a lot of experiments, but this is what we saw. So these cells produce ribbons, and although the cells in the ribbon are short-lived, the ribbon itself lives as long as the mouse, which states that the stem cell is indeed live for 2.5 years. Another version of this experiment, rather than just turning the stem cells blue, which is about probably 15 of these stem cells in a gut, we obtained from Jeff Lichtman, Harvard, these the 4-color brain bow reporter, where 2 colors in inverted pairs are oriented here. We gave it a roadblock, strong promoter, knocked it into the ROSA locus, and this now allows us to stochastically activate one of 4 colors in the heterozygous situation, or one of 10 colors in the homozygous, 10 false colors in the homozygous situation. And I'll draw your attention to the fact that actually we can take a red cell and then flip it over by these inverted LOXP sites and turn it blue at the second retracing event. And these would be the predictions, and that's actually what we saw. About 15 of these cells in the crypt. It's a little bit like an anarchy. They don't divide asymmetrically, you just divide. They compete for space. We know that the Paneth cells, these large cells, make all of the growth. They make Wnt, they make EGF, they have the notch ligands, and every cell that touches a Paneth cell is a stem cell. And by neutral competition, eventually one will, by chance, win, and our animator was hoping that we would see something like this inside our mice, and he agreed he was a little over the top here. But these are the pictures that we get. So the muscle layer is here. This would be one villus, another one. And you can see, I told you, multiple crypts center around the base of a villus. And you can see that the cells in different colors stream out of these crypts and on an enlargement. So after about 3 months, every crypt is now monoclonal. This one's red, blue, yellow, green. And there's very little mixing of these cells while they move up to the tips of the villi. Now this experiment confirmed that Leblanc was true. His crypt-based columnar cell, through these intermediate stages, can make all of the stem cells of the intestine. And we've since done very similar experiments in hair follicle and kidney and liver and lung, and we invariably find that these stem cells are driven by Wnt, they have LGR5, and using lineage tracing, they really are the stem cells of those organs. As another bit of history, Paneth, Austrian pathologist, published a paper in 1887 where he described the Paneth cells, so these large cells. I had this paper on my iPad and I magnified one of the figures, and then I realized that he had seen Leblanc cells here and here, and he'd even put an S there. And I thought that the first use of the word stem cell was Tillem McCullough in the 50s. So had I discovered the real discoverer of stem cells? And then I read, this is all German, so now if you're a PhD student in the 19th century, you don't write in Latin, but you write in German. It says schmale Zellen, which means small, narrow cells, I guess. So he could have been world famous, but at least he discovered the Paneth cells. So there are many unusual things about the stem cells in the crypt. They don't divide asymmetrically, so the textbooks will tell you a stem cell is quiescent, divides very rarely. These cells divide every day. Symmetrically, they then compete for space, and they do this for the lifetime of a mouse. And this would be about 1,000 cell divisions. In our gut, it would be about 25,000 cell divisions. We think they do this. They apparently managed to keep their genome intact. And Toshi Sato in my lab then decided, against the dogma, would it be possible to actually take one of these stem cells? We knew quite well what the growth conditions are for these cells in vivo, and could we mimic this and actually grow these stem cells in vitro, where all of us believe that it's not possible to grow non-transformed cells. It actually turns out to be very easy. A cocktail of 3 growth factors in the mouse, 4 or 5 in humans. We picked our spondin at a time that we did not know it was the ligand of our LGR5. I just showed you this in a little animation, because we knew we had to activate the Wnt pathway. And Wns themselves are very awkward proteins because they're lipid modified, difficult to produce, they stick to plastic. A very lucky pick here. Epithelial growth factor, for all epithelia is always good. And then we knew from an old experiment we had to block BMP signaling, transgenic mouse that we made a long time ago. And we do this in 3D. So we take a single cell from a mouse or human gut in this cocktail, over the next 2 weeks or so grows out in a fairly chaotic structure. If you then break it up, and we passage them every week, we have now done this for 3 or 4 years, they make these rather symmetrical structures that Toshi has called mini-guts. And this is a different way of looking at these things. There is a central lumen, there's multiple crypt-like structures, stem cells are here, pennant cells are here, transient amplifying cells are here. All of the cell types that the normal gut has are actually sit and surround this central lumen. So in some way, a single stem cell on its own in this very simple cocktail of 3 growth factors can recreate essentially a gut. We've since done this for various tissues, so we can do this in human and mouse. We've now come up with conditions for stomach, small intestine, colon, liver, pancreas. No, you can read this. Very importantly, even after a year, when we look at the chromosomes, they're normal. When we look at structural aberrations in the genome, we do not detect any. Telomeres are long. The only thing is we do see point mutations, but the rate at which they occur in a deep sequencing whole genome analysis is about the same as you would see in the germline. So they're genetically extremely stable, phenotypically extremely stable. Small intestine stays small intestine, will never become colon, will never become stomach. And by all means, we think they are representative of the in vivo tissue. We've been thinking about applications, of course. We're also asked to say a little bit about the future of our own science. 2 applications have come up. One would be regenerative medicine, obviously using adult stem cells like bone marrow stem cells have been used for 30 or 40 years in hematology. And the second one would be to more immediate to use these systems to do some disease modeling in the lab. Here we collaborate with Mamoru Watanabe's lab, Toshi Sato's, my postdoc's previous lab in Tokyo. Toshi sorted a single stem cell from an RFP mouse from the colon, so the Dutch cell is red. It makes red organoids. He grew them up to about 100 million, so from 1 cell to 100 million from an adult mouse. Froze them, you can freeze and thaw these things quite easily, FedExed them to Tokyo. In Tokyo, Watanabe's lab had prepared mice with DSS, which causes superficial ulcers on the inside of the colon. It's a colitis model, experimental colitis model. They infused Toshi's organoids through the anus into about 30 or 40 mice. Close the anus with a little bit of super glue for a few hours so that the organoids that have their basal sites on the outside could adhere to the exposed laminins and collagens in the ulcers that these mice develop. These organoids will actually land there. They will not adhere to the normal epithelium. The moment they land, they open up and they actually, like a living band-aid, will seal these wounds. And the mice that we do give these organoids do much, much better than the control mice. Now, some of you might have noticed this is a movie. This is not a, it's not the real experiment. Here you do see the data. So histologically, our pathologists cannot see where the transplanted tissue is and where the normal is. Fully integrates. By all means, it is normal tissue, and only when we look under the confocal we can see the RFP. So this is the Dutch single stem cell derived colon material in a black Japanese mouse. We can follow these for now 6 months, and we see no changes in terms of exhaustion of the stem cells that people might predict, or transformation, adenomas or something like that. So it looks like they're really normal, in line with the fact that we see no genetical changes in these cells over time. Disease modeling in this system, cystic fibrosis, you probably are aware that in cystic fibrosis there are mutations, various mutations in the gene that encodes a chloride channel. The most common one, F508, is a small deletion of one amino acid, which results in a temperature-sensitive version of this channel. Importantly, also the wild-type channel can be opened. So here it's closed, but can be opened by raising cyclic AMP, for instance, with forskolin. Now, if you do this, so if you take small biopsies from a kid, a rectal biopsy, which is very non-invasive, and we actually grow them, it takes about a week to have enough material. If we stain them in green, then add forskolin, in a period of about 2 hours, they very rapidly swell up. So you can have computers looking at the surface area, and it turns out to be a fantastic functional assay for a CFTR. When we do the same thing from a cystic fibrosis patient. So this is one of those patients with an F508 mutation. At 37, we see nothing. If we grow them at 27 for about a day, now we apparently bring the channel to the surface of the cells, and now they actually, I don't show it here, but now they actually open up. So this is a very nice assay, functional assay that we have a readout in about a week directly out of a patient. And actually, we are now collaborating with several pharma companies who have experimental drugs that they find very difficult to predict which one will work for which patient. And it looks like we can use this assay to help streamline trials with these drugs. To summarize this, so normal organoids, when you raise cyclic AMP, will swell. The genetic defective ones, the cystic fibrosis, will not. Can we repair the gene? And here you see the DNA coil with the 3 base missing. So we hope, this actually paper we published last year, using Cas9 CRISPR, that we can use a double-strand break, which is quite easy in these organoids. And then by providing a homologous recombination template, I think the biochemistry is a little bit more complicated than what the movie shows here, but we could replace the mutation by the wild-type insertion of the 3 bases that were missing. We then have a single stem cell that has this mutation. We included a puromycin resistance allele, grow it up into organoids, and now compare them to normal raised cyclic AMP. And indeed, the prediction would be that we now have repaired the defect, so they will swell. And the real experiment is here. So this is the F508 patient that does not respond to cyclic AMP, but after we repair its stem cells, you can now very nicely see that these swell. Now, this is probably not a good therapy for cystic fibrosis because it's a multi-organ problem. We've since moved on to do it in liver, where we can grow single human liver stem cells into as much tissue as we want. And I think we just managed to repair the alpha-1 antitrypsin gene in a patient that's deficient in that gene. And there you might think of maybe in a number of years to use this for in vivo treatments. A last application, since we can grow normal stem cells and we see no induction of any genetic change, we also ask, can we grow cancers? Turns out to be easy. The problem is that if we do this, for instance, for prostate, and we do this together with Charles Sawyers, that the conditions do so well that it's very difficult to separate the cancer stem cells or the cancer cells from the wild-type cells. The wild-type cells grow faster. We lose cancer cells because they tend to be genetically unstable, we think, and we see a relatively high level of apoptosis, whereas the wild-type tissue will just continue to grow every day. So we have to find ways to separate wild-type from mutant. In colon, it's very easy because almost all colon cancers have activating Wnt pathway mutations. About 80 to 90%, but Bert will know much better, have APC mutations. We'll see TCF7L2 fusions, so TCF4 fusions, R-spondin fusions. You'll see RNF43, the E3 ligase I told you about, is mutant in some of these colon cancers. Normal tissue will grow in the complete mix. It will not grow in the mix where we leave R-spondin or Wnt out of the medium, but the colon cancers will happily grow without Wnt. So we've been building up a large panel. We're now probably up to about 30, where we have normal epithelium growing as well as tumor tissue. Matthew Morrison at the Dana-Farber and the Broad has been sequencing these tumors, and Mike Stratton has his robotized, at the Sanger, has his robotized platform to screen them for drug sensitivities. Sequencing effort turns out that actually we capture the entire, if you would call this, the richness of the disease, so we can grow the cancers of every patient we get our hands on. We get all sorts of combinations of mutations. There's a fair number of colon cancer cell lines, but we already now have unusual combinations that are interesting from a drug targeting perspective that are not represented in these cell lines. And they're screenable. You can see that here. So Mike Stratton had these papers in Nature where he collected all existing cell lines, and on a robotized platform, random against a large series of well-defined drugs. This is also doable with some changes with these organoids, so it's a simple ATP readout. And for instance, we noticed that patients that have a wild-type RAS are sensitive to these drugs here, whereas the KRAS mutant patients are not. So we think we can really use this platform as an intermediate between we've heard a lot about this, the sequencing efforts, that are ongoing in human cancers, and the only functional readout at the moment we have is the sensitivity to drugs and trials. So we think this technology that we now have going for pancreas cancer, prostate cancer to some extent, lung cancer, and colon cancer, we think we can actually use this technology in between the genetic data and the patient data. And because these organoids are amenable to any molecular technique that you'd like to use, we think they're going to be extremely helpful in categorizing mutations in terms of in functional terms. And I think with that, a number of people involved, I won't name them all here, but I'd like to thank you very much for your attention.
That was spectacular. I love all the animation. Okay, questions? This is Hans' PhD student.
I'm reversing the roles here. Given that the intestinal stem cell seems like a special beast, do you have any idea how it keeps its telomeres long? Like how is it still a normal cell, but it somehow kept this longevity and yet go through so many cell cycles?
Well, if we look carefully at the program that's activated by Wnt are spondin, so it's not sort of the general Wnt target genes you'd see during development, but these very strongly augmented Wnt targets that you activate with very high levels of Wnt in stem cells. It's essentially is a program for cell renewal. So TERT is there. So TERT is a direct target, as published by Rolf Kemmler recently in Science, of beta-catenin and TCF. There's a lot of DNA repair genes. Bird published that MYC is a target gene. Frank McCormick published that cyclin D1 is a target gene. So this whole program that's driven by Wnt in stem cells is apparently meant to allow these cells to self-renew forever. Unlike the hematopoietic system, we don't see exhaustion. So a pathologist looking at the gut cannot see how old the gut is, which is very different for the bone marrow or for the skin, for instance. So we don't think there's an inherent clock that tells a cell that it's old and it should stop dividing. At least these cells tell us that and in culture we can take liver or we can take lung or pancreas, where there's normally very little proliferation, but in culture they'll do exactly this. We can culture them for 2-3 years with no changes to the overall changes to chromosome structures and a minimal amount of point mutations. Importantly, also, I didn't say this, but importantly, when we look at the point mutations where they're randomly scattered through the genome, they are fewer in the reading frames, in the coding sequences. So it looks there's some sort of a selection mechanism against mutations there, or maybe the transcribed regions are better repaired than the non-transcribed regions in these cells. So it's really I think it's really against any dogma that we have learned about the hematopoietic stem cells, for instance.
I want to ask a question about these adult stem cells. Have you tried to turn any of these into neurons?
It actually is impossible without playing around with their, we'll hear more from Shinya, without playing around with their transcription factors to turn them into another fate. So we can turn small intestine into stomach, but we have to delete CDX2. Okay. And then they will die in the small intestine, but they will survive in the stomach. Yeah.
Okay. All right, thank you.